Hello and welcome to another edition of the Omics cast. My name is Alberto Vilella, and today we're going to talk about GenapSys. As usual, you can, if you want to ask questions or contact me for anything, you can follow me on Twitter or you can contact me on LinkedIn. This slide deck is available upon request, and most of what we're going to be talking about today, you could already found out in this next generation sequencing page that I maintain. And we're going to talk about GenapSys today, which is one of the entries in this technology, in these companies that are pushing for technologies to be the replacement of what we currently call next generation sequencing. That's the reason why I call this page next generation. And in this page, you'll see some established players like Oxford Nanopore or Pacific Biosciences, which is in the process of being acquired by Illumina. And there are going to be some smaller ones and some historical ones. So GenapSys is one of these companies, and they've announced recently some more information about their technology, and so we can discuss it today. The next generation sequencing page that I maintain also contains information about the technology that they use, things like clonality, base-by-base readout, base processing, type of termination. GenapSys is a native termination technology, so they don't use base termination, which means this has certain advantages, making it potentially cheaper and faster to run. And it's a company, this is in the logarithmic scale. It's a company fighting for a very competitive market, a market that Illumina is currently dominating, but GenapSys in this plot that shows Illumina and Oxford Nanopore and PacBio is a company somewhere here in the middle, wedge between these big ass companies that have a lot of money and a lot of power to invent new technologies. So GenapSys is one of these players that's trying to do something different, trying to push for products that will improve the next generation sequencing field. So there's been a recent paper, that's the reason why it's a good time now to talk about GenapSys, where they describe, it's a preprint, it's in bioRxiv, they describe where they are now, essentially what they have on this small scalable platform that uses electrical detection of single base incorporations. I think all of the people in this preprint are GenapSys employees. And as you can see, there's quite a lot of people there, and it means the company has a good amount of people working on this. So in the abstract, I've highlighted here two of the main aspects of what they claim they can do. A platform capable of generating 1.5 gigs of data with an accuracy of 99% raw, rates of up to 175 base pairs, although this is up to, this is not probably the median or the mode. And then they say that they are able to identify low frequency mutations at the expected allele frequencies, basing their claim on reference panels that they've sequenced through their technology. So their technology is essentially this lunchbox-sized instrument that has a little open at in here that you can use to put the Flowcell in, or whatever you want to call it if it's not a Flowcell, the chip. In terms of what happens before you load the chip, not very different to what other companies have already tried and proven successful, which is a library prep where the insert here in this brownish color receives some adapters on both ends, and then they're stitched together, and then they amplify each of these molecules in what they call clonal amplification using these nanoballs. I don't think they call them nanoballs due to commercial reasons, but essentially beads that, starting with one copy of the molecule, they can amplify into multiple copies. and then they place them on the chip, and the chip has this mechanism to read out the electrical current through incorporation of bases, and then this is read in the chip and can be analyzed and produced either on the cloud or locally. The FASTQ files, the files outputting the sequence, the DNA sequences, and do some other analysis here. They mentioned variant reporting. So in this figure, they explain essentially how this works. It's a CMOS sequencing chip containing millions of individual sensors. And one of the neat tricks that they claim they're able to do is that they can load each of these sensors with one bit, one clonally amplified bit. And here they show in the illustration how each of the bits is nicely placed and centered around the chip. And so then they say that the sensor here is able to measure what happens at the molecular level in this hairy ball of DNA molecules attached to the bit. And then that's how they process the information. And here they show a plot of essentially showing changes of signal in the electrical current. For those of you that haven't bumped into this preprint and already knew something about GenapSys, you probably have already read Nava Whiteford's blog post about them. And if not, I recommend that you do in 41j.com/blog. There's a blog post about GenapSys that shows some of the details of GenapSys, the company, their patent applications, how they applied for patents. And they show their instrument and the chip, and here a cartridge with the reagents. Very similar in many ways to what other companies are doing out there. And they show this scan of the bits on the chip with a high occupancy. So each of these white dots is a bit, and you can see it's neatly arrayed on the chip, right? So they must have a way of essentially being able to place these bits so that they land on top of each of the sensors, and then they can capture the electrical signal for each of those. Further down in the paper, they describe essentially the 2 or 3 steps in which this happens. First of all, they load the bits. Bits were loaded onto a sequencing chip, which has an array of sensors. It's capable of capturing 1 bit. And so that's very important that they can do this well, meaning they can load the chip with as many bits as possible, but that each sensor only gets 1 bit. Then they inject the polymerase and incubate for 5 minutes to allow for binding to the DNA templates. It's surprising how polymerases, we think in this molecular biology part of what happens in our cells, that we think it's so efficient, and it's not actually. So if you were to measure what can happen to each of these polymerase molecules when they're floating around the chip and trying to find a bit to land on, it's not very efficient. So they say they have to incubate it for 5 minutes to have a good occupancy, a good binding of polymerase to bits ratio. And then they say that the sequencing begins with electrical calibration of the chip, whatever that means for their purposes, followed by the serial injection of individual nucleotide buffers. For each injection, the chip was incubated for several seconds to allow for nucleotide incorporation. So they say several seconds, but they don't say how many. And then electrical signal of individual sensors was captured. And then the chip was washed, and this process was then repeated using a different nucleotide. So they essentially say you have to inject the nucleotide buffer, incubate for several seconds to allow for nucleotide incorporation. Presumably this incorporation is then captured in the electrical signal, wash it away, and repeat again with a different nucleotide. So this is very reminiscent of what Illumina has done for a long time in their four-based 4-dice technology, inject each die, see what incorporates, measure it somehow, wash it away, inject the next nucleotide, and so on for the next cycle, and so on. And they say that to sequence 150 base pairs, approximately 250 to 300 nucleotide flows were required. So presumably they don't do it synchronously. I mean, they couldn't do it synchronously 4 bases at a time, which means they assume that in 200 or 300 flows, all the reads in each of the bits have already reached 150 base pairs, which, it's a good approximation. How is this technology similar or different to other technologies? It's got similarities to DNB-Seq, the MGI-BGI technology, in the sense that they have to take these balls and grow them somehow and then make them land on these chips, on these sensors, on these wells in the chip. And this is a screenshot of what the DNB-Seq technology claims to do. Very small wells, in that case, 300 nanometers, although they're quite distant to each other, as you can see here. And the DBN technology, they also arrays them in this grid pattern, as you can see here. So similar to what GenapSys is doing. Compared to Illumina, they are landing bits where they've already amplified things. And the Illumina technology for the pattern flow cells is not like that. It's based on what they call ExAmp, which is simultaneous seeding and amplification, which allows Illumina to maximize well occupancy and monoclonality. Because the amplification rate is much faster than the seeding rate, the templates are excluded from occupied wells, meaning one molecule lands on a well and then very quickly amplifies. And before any second molecule can land on that well, the well is already occupied, and then the next molecule cannot land, or if it lands, nothing's going to happen. It's going to just detach and go away. But as you can see, Illumina flows the unique molecules into the empty wells, and then with the ExAmp technology, they maximize well occupancy, which is different to GenapSys and the DNB-Seq technology from MGI-BGI. So back to the GenapSys preprint. Here they show some sequencing statistics, some what's called in the field GC bias plots. They're called GC bias because they'll show you if you have bias in the in the reads that you sequence. Essentially, if you don't have bias, meaning if you sequence exactly the same reads that you flow in at the same rate as they come, then you should have a series of dots here around the 1.0 mark, right? So for example, here in blue, GenapSys does relatively well from about 10% to about 40-50% in this Campylobacter jejuni genome. It's a bit noisy here and here, where it underrepresents molecules at high GC and high AT, and it overrepresents a little bit at around 40%. In E. coli, here in green, they show Illumina much better than GenapSys, where at around 55-60%, it drops down substantially, right? So it's underrepresenting massively this high GC fraction of the E. coli genome. And for this other one, Illumina has this kind of triangular shape, which is not perfect. It underestimates the low and high GC and overestimates the mid GC. But GenapSys is even worse because it over sequences at the low GCN and under sequences at the high GCN. So this paper, even though compared to Illumina for the plots they've chosen, not a perfect series of genomes here, these 3 genomes, GenapSys does not do perfectly, and in some of them does worse than Illumina. In terms of frequency of the variants detected, they show this table with the genes and the variants inside the gene, what the expected frequency is from this reference material they used, what Illumina gets, and what they get. So the one I highlighted here is this one that was expected to be a 9% frequency, and GenapSys only gets it at 3.8%. Illumina gets closer to 6.6. The other ones a bit noisier, but not significantly noisy. I mean, this one maybe a little bit. And when they plot these against the expected allele frequency, it doesn't look that bad, simply because of the effect of plotting things that are very close to zero, for example, here. So I mean, it's not terribly bad. It's something nobody would worry too much about. So the conclusions and the remaining questions for this GenapSys instrument or technology is that it's a small, low-cost instrument, currently somewhere in between the Illumina iSeq instrument and the ONT MinION Flowcell, both in size and in cost to the people that buy it. It produces short reads, relatively high quality, similar to Illumina. As far as I can tell, they're all single end reads. They don't do pair end like Illumina does. They're aiming initially at targeted panels, maybe exons, and they're aiming at growing the chip and having more wells or more sensors in the chip and increase the throughput. But then the questions would be, how difficult is the sample prep? Can it become a very simple sample prep that can enhance this as a product altogether and be a point of care device? Imagine you go to the doctor or to the dentist or anywhere where it needs some DNA sequencing, and they can take a sample from you and do a very quick library prep, load it on this lunch-size box machine that's sat somewhere in a table in the doctor's office, wait a few minutes, maybe an hour, and then by the time you're home from your visit, you'll receive a report of what the doctors found out, and maybe they make you come back. Or if this is the bedside, similarly, you're in hospital and then the machine is loaded with your sample, and within an hour it gives you an answer, and they base the treatment based on that, rather than having to extract the sample, send it to a centralized lab that has a queue of samples waiting for it, and only a certain number of big expensive machines to use, and then your sample is in the queue and it has to wait for a bunch of other samples to be ready, and the library prep takes hours, etc. So point of care is a big plus for any technology that can make this immediate, quick, with a low cost instrument, etc. And then, so final questions for GenapSys: how will the throughput go up? Will they be able to compete with existing technologies to do a 30x whole human genome? And how fast will it be if each incorporation of the nucleotide only takes several seconds? We don't know how many, and they only need to do 250 or 300 of this. Can they do a human genome at a decent cost in a matter of minutes or an hour? And this will compete with other technologies at a fair standing. And as usual, here's my contact details, and you can contact me via Twitter or LinkedIn and ask questions about the world of genomics, omics, sequencing technologies, whatever you fancy. Thank you very much for listening, and I'll catch up with you in another episode of the Omics Cast.